Welcome back to Part 3 of Chapter 2, Anatomy Physiology 1. We left off talking about water and hydrophobicity, and I wanted to talk about some unique properties of water. Water has the ability to dissociate into a positive hydrogen ion and a negative OH radical. Now, depending on what is in the solution with the water, something may come along and take up the OH, leaving a lot of hydrogen free in the solution. The more free hydrogen that there is in the solution, the more acidic that we say it is. And if you want to go and learn about the logarithmic scale, you're welcome to do that. But take my word for it, if you have a lot of hydrogen, then your pH is anywhere from 0 to 1. And if you have very little hydrogen, then your pH is going to be all the way over to 14. So this is your pH scale, and it's the logarithm of the hydrogen ion concentration. Some of the things that are very acidic would be stomach acid, lemons, vinegar, which is acetic acid. Oddly enough, bananas and tomatoes are very acidic. Coffee. When you get into this range, you're talking about things that are neutral. So they're not acid, they're not base, they're just neutral. And you're going to have the same amount of free hydrogen as you do the OH radical. And then once you leave this range, milk and water and eggs, now you're getting into household bleach, ammonia, and oven cleaners. And then if you've ever heard your great-grandparents talk about lye soap, then you're talking about sodium hydroxide. Here's a slide that kind of summarizes what I just said. If you take the hydrogen away from the water, the oxygen is going to hang on to the electron. So you're literally just going to have a free proton. So you're going to have a positively charged hydrogen ion. The OH is the oxygen, the other hydrogen, and because the oxygen kept this hydrogen's electron, it has an extra electron, more than what it ought to have, so it is a negative ion. If you have an equal amount of hydrogen and hydroxyl, then you are neutral. If you have more hydrogen, you're acidic. If you have more OH, you're basic. Most everything in your body is close to 7, which is neutral, but some exceptions would be your urine and your stomach contents, because your stomach secretes hydrochloric acid. For those of you who enjoy math, here's the formula. The pH is negative log of the hydrogen ion concentration. So if the hydrogen ion concentration was 10 to the minus 3, then that would be negative log 10 to the minus 3, which would be 3. So that would be very acidic. Some other measurements that you need to know besides the acid and the base measurements are: how do you weigh medicines? How do you weigh test results? So if you're giving somebody an IV, you're going to have something in the water. You don't just run water into a person because it would kill them. And we'll learn out why a little bit later on. So you're going to put like 0.9% saline, or you're going to put a sugar solution. So sometimes they're running an IV of glucose or dextrose, as it's really called. Sometimes there's ringers because the person needs ions and things like that. So we often use just a percentage because most people understand percentages. One of the other ways that you measure things is per liter or per deciliter or per milliliter of blood or urine, whatever it is that you're studying, stomach contents. So you've probably been told that your cholesterol should be below a certain level of so many milligrams per deciliter. I'm going to tell a cautionary tale here that I think is kind of funny. At one of the universities I was teaching chemistry, we had some young ladies who decided rather than buy the hydrogen peroxide to be bleached blondes, that they would just borrow some from the chemistry lab. And what they didn't realize is the hydrogen peroxide that you buy in a store is 3%. So there's 3% hydrogen peroxide and 97% water. But the stuff that they borrowed from the chemistry laboratory was 30%. And it was very easy to tell which were the girls who borrowed the hydrogen peroxide, because at that concentration, it literally ate the hair off their head. So they were easy to pick out. Since we've been talking a little bit about chemistry, let's throw a little physics in there. We need to talk about energy. So we talk about ATP energy and work. This is one of the things that when I teach physics, people get really upset about, because if you were to walk over to a wall and push on the wall and push until you were exhausted and you're sweating and your muscles are quivering, you will have expended energy, but you will have done no work because you did not move the wall. So work involves moving something. So no movement, even though you used up energy, it's not considered work. So that always frustrates students. We generally categorize energy as either potential energy, which means you could do work, you could move things, but at the moment you're not doing anything with the energy, it's stored up. So that's potential energy. The other energy is the energy of motion, and we call it kinetic energy. So in the chemistry of motion, you have chemical energy, the energy of heat, and electromagnetic energy. We spent some time talking about how atoms are bonded together. We talked about ionic bonds and covalent bonds, which is a sharing of electrons. When you are putting atoms together, we call this synthesis. And when you're tearing molecules apart, we call it decomposition. So you are familiar with that term when they talk about a body decomposes after death. The chemicals inside the body, the proteins break down, the lipids break down. And then you have exchange reactions where you just basically swap one ion for another ion. So if you had sodium chloride and potassium chloride, you could just switch the sodium and the potassium. Here's one example of an exchange reaction. In your stomach, you have HCl, hydrochloric acid, and you have sodium bicarbonate that is made by your pancreas to help neutralize the stomach acid so it doesn't eat a hole in your intestines. So when the hydrochloric acid and the sodium bicarbonate mix together, then you get just plain old table salt, NaCl. So there's the sodium from this one exchanging with the hydrogen here. So now you have NaCl, and this hydrogen goes over here, and now you have two hydrogens and CO3. And this is carbonic acid. And if you look at that carefully, you can see that this can further decompose into water, H2O, and then you have CO2, which is carbon dioxide. So this can break down further into water and carbon dioxide. When you eat something that's starchy, for example, pasta or rice, then in your mouth you have an enzyme called amylase, and it just starts taking the sugars off of the starch. And now you have free sugar, free glucose that you can use to keep the brain going, to keep your muscles going. So they call this a complex carbohydrate because you can break it down into smaller units. So here's one of the more common decomposition reactions that happens every time you eat anything that is high in carbs. In one of our upcoming chapters, we're going to spend some time talking about how you can take the different amino acids and you can pop them together by making them share electrons, and you can end up with a protein. So we're going to learn that you can synthesize proteins from amino acids. A lot of reactions are reversible. So you can build up proteins or you can break the proteins down. You can exchange one molecule for another molecule, one atom for another atom, and then you can exchange it back. So this is constantly happening in the body, that we do reversible reactions. And they usually indicate reversible reactions by having an arrow going in both directions instead of just an arrow going in one direction. So I mentioned the carbonic acid. Here you have carbon dioxide plus water makes carbonic acid. So if you've ever drunken any fizzy drink, Coke, Pepsi, Mountain Dew, what you're drinking is carbonic acid. And if you leave your Coke or Pepsi or whatever it is that you're drinking out in the open for very long, what's going to happen is the carbon dioxide is going to outgas. It's just going to go off into the air because it is a gas. So as these things dissociate, the carbon dioxide leaves and your drink goes flat. It isn't acidic anymore and it doesn't taste very good. Now, if you just took some carbon dioxide and bubbled it back into your Coke or your Pepsi, it would taste just as good as it did before when it was all fizzy. So you can re-fizz it by putting in carbon dioxide. So you've been performing chemistry all along and you didn't really stop and think too much about it. But if you want your sugar to dissolve in your tea, there's a couple of things you can do. One of them is stir it. So you increase the surface area touching. You can increase the concentration of the reactant. You can heat it. Again, with that example with the tea, trying to get your sugar to dissolve in your tea. If the tea is warm, it's very easy to stir the sugar in. But if you've already got the ice in your tea and now you're trying to dissolve the sugar, you find you have to stir and stir and stir to get it to go into solution. Another thing that will cause chemical reactions to occur more quickly is to use a catalyst of some sort. So in your car, you have a catalytic converter, and the reason that they are so expensive and then why people are stealing people's catalytic converters is because they have a precious metal in there. Inside of the catalytic converter of a truck, you've got about 15 grams of platinum. So if you've ever bought platinum jewelry, then you know what I'm talking about. They're very expensive. Enzymes also act as catalysts. They hold the reactants near each other so that you can encourage them to share electrons. Or if you want to tear it apart, you have the molecule held by your enzyme, and the enzyme flexes and literally pulls the two atoms apart. So you can do synthesis or decomposition using enzymes, and that's what we do a lot in our bodies. So of course we have to have another name for decomposition reactions. So they are called catabolic reactions. And catabolism is the tearing down of things. And I don't have any trouble remembering the difference between catabolism and anabolism, which is the building up or the synthesis, because I just see a cat shredding pretty much everything in a room. In case you need a visual for catabolism, here's a cat clawing this furniture to pieces. Another set of terms that you need to know is oxidation and reduction. And they always seem to like to ask this question. So I'm just going to see if I can help you put it in your head. So if you go back and remember, oxygen has 6 electrons in its outer energy level, and it's looking to take 2 electrons. It's looking to gain 2 electrons. Then if something is oxidized, it's gaining electrons. Let me say that again so that I can make a distinction. If you are talking about oxidation, here is where a molecule will give up its electrons, oftentimes to oxygen, and energy is released. The molecule is oxidized when it loses its electrons. So oxygen is the one that's going to be taking the things, so it's going to be the oxidizing agent. So there's words that have almost opposite meanings. The molecule is oxidized when it loses its electrons, and the oxidizing agent is what takes the electrons. And it's oftentimes oxygen, which is why they call it oxidizing agent. So I apologize that it is confusing, but you do need to know the difference between oxidation and reduction. So the chemical reaction in which a molecule gains electrons, like oxygen getting those electrons, is reduction. So the molecule is reduced when it accepts electrons. Oxygen accepts the electrons, so it is considered to be reduced. But the reducing agent is the molecule that gave the electrons to the oxygen. So in the case of water, hydrogen is the reducing agent, and oxygen is reduced. Instead of saying oxidation and reduction, since they both occur at the same time, one is oxidized and one is reduced, they call them redox reactions, oxidation, reduction reaction. Organic chemistry is a study of things that have carbon. That used to be the definition, but then people started saying, well, how about carbon dioxide? It's not a protein, it's not a lipid, it's not part of a living being. It's just a gas that's given off as a waste product when we exhale. And so they said, okay, so we're going to change the definition of organic chemistry to things that have carbon and hydrogen. So that's the working definition now. And one of the things that astounds me is you can take an entire year, you can take 2 full semesters of organic chemistry where you're just studying carbon. That's astounding. Now, we take the carbon compounds that are in our bodies and we break them into 4 macromolecule or huge molecule groups. One of them is carbohydrates, one's lipids, one's protein, and one is nucleic acid. So those are your macromolecules made from carbon. If we go back to our periodic table and we look at carbon, it's element number 6. It has 2 electrons in its inner energy level and has 4 valence electrons in its next energy level. And it is in column 4. IV is the Roman numeral 4. When it's bonding, it can either give up 4 electrons or it can take on 4 electrons. So I think I mentioned that it can bond with pretty much anything on the entire periodic table with the exception of the noble gases. And they don't share electrons. So this makes it very versatile, and you're going to find it in pretty much every molecule in your body. So we'll be talking about the 4 macromolecular groups in the last part of this chapter. So, one of my favorite episodes of Star Trek was when they went to a planet and people kept mysteriously dying. And if you remember, they were always having their phasers and they were blowing up rocks and they were making pathways to get from one place to another whenever they would go onto a planet. But what they didn't realize on the planet that they happened to have landed on is instead of being carbon-based life, they had silicon-based life. And if you look, it also has 4 electrons in its outer energy level. So all the compounds that we have that we make out of carbon on that particular planet, they made out of silicon. So the rocks were actually alive. And so when they were running around with their phasers, blasting holes in rocks to make pathways, they were actually killing the people that lived on that particular planet. So if you have time, I recommend you watch that episode because it's really cool to watch Spock mind meld with a rock. Here is one of the simplest carbon molecules that you can make. So here you have a carbon and you have 4 hydrogens around it. This is the one way of drawing the image. Here's a space filling model right there. And here is a 3-dimensional representation of it. So they're showing you that it actually is like a pyramid. Here they're trying to show you all the bond angles. What's interesting to me, carbon with 4 hydrogen is methane, and that's what we call natural gas, or to be crude, that's a fart. Here's a picture showing some of the things that you can make with carbon. Carbon can combine with itself. It can share electrons with another carbon. In this particular case, you have propane. So if you have a propane tank or you heat something with propane, this is the formula for that. 3 carbons and 8 hydrogens around it. Butane, if you have a butane lighter, it's 4 carbons and 10 hydrogens. But you can also take propane and put the carbons in a triangle like this and put the hydrogens like this. And so you have cyclopropane, or propane that's kind of in a circle. You can have isobutane. So here's butane like this, but you can branch it off. There's 3. So you still have 4 carbons, but you have all of these hydrogens. So now you can see how you could get into spending 2 semesters talking about all the things that you can do with carbons. You can make rings, ring compounds. So anyway, if you have the opportunity, I would take organic chemistry because it's fascinating. We've been talking about individual atoms bonding with individual atoms, like hydrogen combining with oxygen, sodium combining with chlorine, sharing electrons. But there are certain groups of atoms that kind of hang out and are adopted or bonded as a group. So we talked about if you have the water and you split it, you've got your hydrogen, and then you've got your OH group. That's called a hydroxyl group. And it will bond with something as a group. Methyl is a carbon with 3 hydrogen, and it will bond as a group. There's a carboxyl group, which is a carbon, 2 oxygens, and a hydrogen, and it bonds as a group. So it's kind of like a clique, It's like if you're going, I'm going. The amino group is a nitrogen with hydrogens, and a phosphate is phosphorus with oxygens attached to it. So we're going to talk about the phosphate group when we talk about ATP. We're going to talk about it when we talk about the phospholipid bilayer that makes up the cell membrane. We're going to talk about the amino group when we talk about amino acids. So that's how they get their name. And of course, we're going to talk about hydroxyl groups because we're going to find that water plays a huge role in anabolism and catabolism and oxidation and reduction. Here's a slide showing you the hydroxyl it makes your sugars and it makes your alcohols. Methyl is going to be part of your fats, steroids, part of your amino acids. But one of the things that we're really going to get into when we get into genetics is if you add a methyl group to a gene, you can turn the gene on or turn the gene off. So we have a whole new field that has just been discovered called epigenetics. And the methyl group is super important in epigenetics. The carboxyl group I was talking about, the carbon, 2 oxygens, and a hydrogen, you're going to find that in your proteins and your sugars. I already said the amino group you'd find in the proteins and the amino acids that make up the proteins. And then your DNA and your RNA is held together with phosphate groups. So you have a phosphate backbone that we'll talk about. So I'll not ask you to write out these structures, but you should spend some time and look. Methyl is a carbon with 3 hydrogen, carboxyl. Amino is nitrogen with some hydrogens. Phosphate has oxygens and hydrogens attached to it. So I mentioned the 4 macromolecular groups, and I told you if you forget them, just look on a candy bar or a bottle of drink of some sort. And by law, you're required to tell how much protein, how much carbohydrate, and how much lipid. You don't have to talk about the nucleic acids, though. And one of the things that I hope you had the opportunity to do sometime in high school was to take some Elmer's glue and mix it with borax, which is something that you use in your laundry. You can buy it at anywhere you buy laundry detergent. If you mix these together, it takes the protein that's making up the glue and it forces it to share electrons. And so you go from having glue to having slime or oobleck, or whatever you want to call it. And you can actually take it and make it into a ball, and the ball will bounce, and you don't get sticky all over your hands. So this would be called a polymer, or mini units. So you take the protein that made up the glue, and you daisy chain it, and then you cross-link it, and you end up with a solid instead of liquid glue. This slide and the next slide are going to show you dehydration synthesis and hydrolysis, which breaks things up. So I mentioned that water can dissociate, and you can have hydrogen, and you can have the hydroxyl, the OH group. Well, if you are putting 2 things together, like 2 sugars together, what you're going to do is you're going to pull one of the hydrogens off of one of the molecules, and you're going to pull the OH group off the other molecule. That will go off as water, so it just goes off. And now, because you pull these 2 off, you force the 2 molecules to share electrons together. So you are condensing or dehydrating, or you're doing synthesis, all of those words are correct. So, but it is called dehydration synthesis because you are removing an OH and an H, and they make water, and now you stuck these two molecules together. So in the case of starch, a little bit down the road, I need some energy. I'm going to run a marathon. I'm going to babysit my grandson, something like that, where I'm going to need a whole lot of energy. And what I'm going to do is I'm going to take water and I'm going to do the opposite. I am actually going to I'm going to pull the two glucose molecules apart, stick an OH group on one, stick a hydrogen on the other, and they will go off as glucose. So I had to use a water. So I am using water. Hydro means water. Lysis means splitting. So I can remove a water to put two molecules together, and I can put the water back in and force it to split the molecule and put the OH back on and the H back on. So look at this, think about it, dehydration synthesis and hydrolysis, because these are important for how do we build these macromolecules? How do we put the sugars together, the fats together? So water is very important. So here's the kind of cartoon version. Here is a monomer. In this case, it could be glucose. It could be something else. But you were going to remove one hydrogen off of this one and the hydrogen plus the oxygen off of this one. So there goes your water leaving. And now this oxygen is sharing with this molecule. So here's your oxygen right there. And you removed water. So you force these things to come together. You dehydrated and made a larger molecule. And then here is the same molecule that we had over here, and we're just going to add water across this. So instead of this sharing electron with this and sharing electron with that one, we're going to put the water back in, and now this doesn't have to share with this monomer, so it can drift off and I can use it to fuel my muscles or fuel my brain or make my nerves fire, whatever I need that energy for. So we're constantly doing this, removing water, putting water back in. I won't ask you to memorize too many formulas. Of course, I want you to know H2O. I'd like for you to know CO2, which is carbon dioxide, and I would like for you to memorize this one. C6H12O6 is sugar. So it is glucose, it's fructose, but this is the formula. You take 6 carbons, 12 hydrogens, and 6 oxygens to make a sugar. When we talk about sugars, we talk about monosaccharides. So glucose is a monosaccharide, it's just one sugar. But then you can put sugar, glucose, and fructose together, and you make table sugar, which is we call sucrose. And it is a disaccharide because it is made from a dehydration synthesis joining of a glucose and a fructose. So it's kind of important that you do learn this formula right here, and that you know a little bit about monosaccharides and disaccharides. There are 3 major monosaccharides. 2 of them get together to make up regular table sugar, like you open a package of sugar at a restaurant. So it's glucose and fructose. And then there's a third one that's important, galactose, because some people are lactose intolerant and they're unable to break down the sugar that's in milk into galactose. So they end up getting gas and cramps and diarrhea if they try to eat anything that has milk in it. They eat ice cream, pizza, pasta with the sauce. And here are some of the important disaccharides: the sucrose, the table sugar, glucose plus fructose. Lactose is glucose plus galactose. And lactose intolerant people cannot break this. They can't do hydrolysis to break these two apart. And then maltose is the sugar that you find in grains. So you know that you get a lot of carbs when you eat things like wheat and rice and other grains. And it's just two glucoses stuck together. If you take an advanced chemistry course like biochemistry, you'll be expected to know the structure of these various molecules and how they're bonded. But this is just anatomy physiology. So whenever you see six-sided rings like this, you know it's almost always sugar. When you look at dehydration synthesis and the formation of a large macromolecule of sugar, glycogen is the storage form that animals do. So we take the glucose molecules, stick them together, and then when we need the energy, we just add water, break the bond, and there's a sugar, add water, break the bond, and there's a sugar. Now what's interesting is if you're looking at this in a human, you call it glycogen, but if you're looking at this in a plant, you call it starch. So we both, plants and animals, store glucose. We make it, store it, and we have it ready if we need it. So we can just, instead of having to synthesize a whole molecule, you just add a little water and get your sugar off that you need. Pay attention to the orientation in space of the oxygen right there, because that's going to be important in just a second. Interestingly enough, we can break down glycogen. Obviously, we put it together, we can break it down, but we can also steal the sugar that the plant has saved up and eat the plant and get the starch out of it so that we can get its glucose. But plants have a little sneaky thing that they can do. They can take the glucose molecules and they can bond them together in a way that we don't have any enzyme to break it down. So it's the same glucose that you'd see in starch, the same glucose you'd see in glycogen, but because of the way they bond it, we don't have an enzyme to break it down. And everybody goes, well, let's see, termites can break it down. Nope, even termites can't break this down, but they have an organism that lives inside of them, so then when they eat the wood, then it can break down the wood and turn it into sugar. So we call this form of glucose fiber or roughage. And a classic example that I always give students is, what do you eat? And then the next morning when you poop, you see it and it still looks just like it did when you ate it. And all the students say, corn. I say, yes. This is what cellulose looks like under electron microscope. Here are the fibers. So if you look at a piece of paper, it looks kind of like this. And if you look at the individual sugars that make up this fiber, you'll notice here's a sugar molecule, here's a sugar molecule, but look at this. This bond is sharing the oxygen in up position, this is sharing the oxygen in the down position, and this is sharing it in the up position. So if it was all down like it was in glycogen and in starch, we don't have any problem breaking that apart. But we can't add water across this bond right there and do the hydrolysis, the catabolism. We can't break it down. We don't have anything that can do it. So once upon a time, about 20-something years ago, I was doing a lecture tour in China, and they were wanting to know things that they could do to help feed their people and genetic things that they could do. And so one of the things that I said, it mystifies me. We have an enzyme that we can take out of that organism that's inside of the termite, and the enzyme can easily break this bond. So all you have to do is put the enzyme on cellulose, on roughage, on fiber, and it will break it down into individual sugar molecules. And you could end world hunger right there. And why we don't do this, I don't know. But you could mow your grass, and you take all that grass and put the enzyme on it and turn it into sugar. You could get the bark off of a tree, sawdust, when you cut up wood, and you have all that sawdust. Throw a little enzyme on it, break it down into sugar, and feed the people who are hungry. And as far as I know, people aren't doing it. But one of the universities where I was talking with the people, one of the professors said, that's a very interesting idea and so simple and so easy. So he said, I think that we can do this. But I never went back to find out if they ever set up a factory to turn cellulose into sugar. So carbohydrates are made of sugars, and lipids are fats. And here's some of the examples that we're going to talk about. Some of them we'll talk about in this chapter, but the phospholipids we will talk about more when we get into cell membranes, because we need to know about that. Pretty much, if you have a pulse, you've heard of triglycerides, because they're always honest about Americans have elevated triglycerides, and they want everybody to take medicines to lower their triglycerides. So we're going to find out a little bit more about triglycerides in a second. Fatty acids, whenever you eat anything that is oily, greasy, waxy, you're going to release fatty acids into your bloodstream. And your body's pretty cool about using the fatty acids to make cell membranes, to make cholesterol, things like that. So we'll talk about that. Steroids, your steroids, testosterone, estrogen, progesterone, hydrocortisone, all of those are lipids, and they're actually forms of cholesterol. So I always get tickled when people say, oh my God, cholesterol is horrible, it's awful. And I'm like, well, actually without testosterone and estrogen, you'd be a mess. And we wouldn't be having children and things, because you need those. So anyway, hopefully I'm here to stamp out ignorance. Steroids in normal amounts are necessary. Anabolic steroids that you buy illegally, not so much. So we'll talk about that again a little bit later on. So here are the groupings that this guy who wrote the book decided were the 5 primary types. I'm not seeing cholesterol there. So I kind of disagree a little bit there, but that's all right. How do you know if you have a hold of a lipid, a fatty acid? Well, it's going to be a chain of carbons, and there's going to be one of those carboxyl groups on one end, which was the COO. Remember that? 1 carbon, 2 oxygens, and hydrogen. And on the other end, there's a methyl group. So those are your fatty acids. And there are some essential fatty acids. So one of the things that I talk about is what makes a sensible diet, because everybody that I know of has been on diets, and some of them are bizarre. I think the weirdest one I know of, a friend right now, is on a bacon diet, and she eats several pounds of bacon a day to lose weight, but she has to give up carbs to do it. So anyway, anything that sounds weird like that, not healthy. And people who go, oh, I need to cut fats out of my diet. You know, I just need to eat everything lean. I need to eat just vegetables and fruits and things like that because they don't have fats in them. Well, they end up dying because you have some fatty acids that you have to eat. So if you're going to cut oils and fats out of your diet, you're probably going to miss out on the fatty acids that you need. So keep that in mind. You've probably heard about saturated fats and unsaturated fats. And again, if you look on your candy bar or whatever it is that you're eating, it'll tell you what percentage of the fats that you're eating are saturated, which are unsaturated, and which are trans fats. Well, trans fats have been outlawed, so you're not going to see trans fats. Everything should say 0 when it gets down to trans fats. So what they're talking about saturated is every carbon has a hydrogen and a carbon. So it is saturated with hydrogens, completely saturated. There's no place to put another hydrogen. The unsaturated fatty acid, look, right here, I could put a hydrogen right there, and I could put a hydrogen right there. So this is unsaturated. And anytime you have an unsaturated fatty acid, it puts a kink in it. So this is straight, and this is kinked. And when you have kinks in your fatty acid, it tends to stay liquid at room temperature. A lot of your unsaturated fats come from plants. So corn oil, olive oil, these are unsaturated. And of course, you've seen, if you've ever cooked with olive oil, you know it's a liquid at room temperature. And fats are saturated. So you've got as many hydrogens as it can possibly hold, and it tends to make a solid at room temperature. So if you've seen lard or fat, if you buy a piece of meat and it's got fat on it, if you look at bacon, the fat on that is saturated, and they're usually animal derived. So it's interesting, I was talking with a friend of mine where we're old enough to remember McDonald's French fries used to be cooked in animal fat, and it was delicious. And then people who didn't want to eat animal products, they were thinking if they ordered French fries that they could be vegan or vegetarian. And then they found out that they were actually eating animal fat, and so they switched over to get away from that, and the French fries stopped tasting the same. So anyway, if you're old enough to remember the different taste of the McDonald's French fries versus the way it is now, it's the lack of that animal fat. So let's assume that you eat a Snickers bar, because that's one of my favorites. But you're going to get the oils from the nuts in there and from the chocolate, and you're going to get all that sugar. And what your body's going to do is it's going to do catabolism. It's going to tear down the peanuts. It's going to tear down the fats and the sugars that you're eating. And then your body's going to rearrange them into things that it needs. And one of the things that it makes on the way to making other things is it makes triglycerides. So if you can imagine a sugar molecule, glucose, and it's got 6 carbons, and you have an enzyme that breaks it in half. You add a molecule of water to split it into 3 carbon pieces. So here is a 3 carbon, half of a sugar molecule, and we call it glycerol. So there it is, that's just half of a sugar molecule. And we're going to do a dehydration synthesis. So we're going to do anabolism, not tearing down, but building up, anabolism. We're going to take the OH right here off of this sugar, this half of a sugar, and we're going to take this hydrogen off of a fatty acid. There's my fatty acid, and there's my carboxylic acid on the end of it, my COOH. And I'm going to take that hydrogen off. And if I take this hydrogen off and this OH off, then water will go away, and this fat will be attached to this carbon through the oxygen. And here's another fatty acid, and I'm going to take it and do the same dehydration synthesis, and I'm going to end up with half of a sugar and 3 fatty acids hanging off of it. So a triglyceride is 3 fats attached to half of a sugar molecule. So if you do like I did, when you're supposed to go in and have blood drawn, you're supposed to fast before you have them look at your fats. Because right after you eat anything, you're going to have a surge of triglycerides, because that's just one of the things your body does with incoming sugars and fats. And so they did not tell me I was going to have my blood drawn, and I was not fasting, and I stopped off and got donuts, which is one of my weaknesses, and they drew my blood, and the lady was white-faced. She goes, "Ma'am, were you fasting?" And I said, "No." And she said, "Oh, thank God." She says, "Your triglyceride level is over 1,000. I'm like, oops. Oh, that would be the donuts that I ate. So here's our molecule that we made, our triglyceride. So there's the half of the sugar molecule. Here's 3 waters that we took away when we made the oxygen share with this fat. Right there. Now look at this. These are saturated, but this one is unsaturated. And this one is actually polyunsaturated because it's unsaturated here and it's unsaturated here. Now, you think that it wouldn't matter where the hydrogen was located on this particular molecule, but this is a normal fat, and it's easily digested. So we can make glucose out of it. We can use it to make whatever we need to do, make proteins. We can just use these carbons and pop them off as we need them. So we constantly are recycling, bonding all of the stuff that comes into our body. So we're the ultimate recyclers. So it was in the last few decades that we started looking at trans fatty acids. In the trans, then you're going to have the hydrogens across from each other, as opposed to having them both on the same side like we saw in the other picture. So if you have it trans or across from each other, it is very hard for you to break this down. So it stays in your body. You're not recycling, you're not pulling it apart to make other things that you need in your body. And so they found that the trans fatty acids would deposit in your blood vessels, especially clogging up your arteries and causing strokes and things, heart disease, heart attacks. So that's why we have banned trans fatty acids, because it's just unhealthy. This is a space-filling model of lecithin. It is, here's your 3 carbons, so that's your half of a sugar. And here's one of the fatty acids hanging off here. Here's another fatty acid hanging off. And they've replaced the 3rd fatty acid with this nitrogen-containing group right here. And what this does, you need this in your body to emulsify fats, to break it up into small droplets so that you can absorb it into your body. So you need lecithin. And if you get a candy bar or something, you'll see that they have lecithin added to it to emulsify the fats. So I don't know if you've ever had a candy bar and you left it in the car, or you found a candy bar that you'd put on a shelf and you come back and you find it a year later and you open it up, and instead of that nice dark chocolate, it looks almost like it's got white mold growing on it. and you're like, ooh, yuck, oh, this is so gross. But what happened was the lecithin stopped being able to emulsify the fats, and so the fats that are in the chocolate rise to the surface. And so what you're actually looking at is a layer of fat that is come out and is now floating on the surface of your candy bar. So it's not mold, it's not nasty, but it no longer is homogenized. It's not homogeneous anymore chocolate. So you're actually looking at the fat that formed on the surface of it. I'd still throw it away because it's old, but it won't hurt you to eat it. When we do the immune system, we'll be talking about prostaglandins. It's a form of lipid, but it plays a role in inflammation, in whether or not your blood clots, in the way your hormones act, whether or not you can go into labor, changing your blood vessel diameter. So prostaglandins are fats, or lipids, that play a very important role in our body. So again, people who say, ooh, fats, oh no, we got to cut fats out of our diet, they just don't know. We just don't need as much fat as we tend to eat. Interestingly enough, prostaglandins are responsible for pain. And you think, ooh, well, man, I would definitely like to get rid of pain, but you don't. You need to have pain because it tells you something's wrong. And it sends a signal to the body, hey, something's not right here, you need to come and fix it. So although you don't enjoy pain, it actually plays an important role. And prostaglandins are the ones that mediate the part of the pain. Once you have seen the structure of cholesterol, it'll stay with you. To me, it looks like chicken wire. So you're going to have 4 rings like this. And this is the backbone, like I said, that makes up your hormones. It makes up part of your cell membranes. So if you were to cut cholesterol out of your diet, you couldn't make your hormones, and you couldn't make cell membranes. So you couldn't replace the cells that die. So you just don't want too much cholesterol. I like that the author made a point to say there is only one kind of cholesterol. So when your people talk about, oh, you have so much of the good cholesterol, or you don't have enough of the good cholesterol, you got too much of the bad cholesterol, that is a misnomer, because there's only one kind of cholesterol, and it looks like that chicken wire stuff. But if you have wads of cholesterol that are complexed with other fats, with phospholipids, with proteins, then you have what we call high-density lipoprotein. So high density, there's a lot of stuff there. It's lipids and it's proteins. So it's not cholesterol, but they just decided to call it the good cholesterol. But that's a total misnomer, because good cholesterol and bad cholesterol are the same. What you're looking at is the size of the wad. So the low density lipoprotein does not have as much of the cholesterol, the fat, and other proteins associated with it. So it's smaller. So the theory is, if you have the HDL, the high density lipoproteins, which they call the good cholesterol, it is attractive to the smaller fats that are in your bloodstream, and so they'll stick together. So oils and fats like to stick together. They do not like water. They don't dissolve in water, and so they'll try to move away from the water and kind of hang out with other molecules. So they kind of say the good cholesterol, because it's so big, can mop up or attract a lot of the smaller fats, oils, cholesterol, lipoproteins that are in your bloodstream. And the idea is if you have a lot of HDL, then you should not be depositing the fats on the arteries and causing hardening of the arteries. So HDL, high density, is supposedly mopping up the smaller to keep you from having heart attacks. There are 20 different amino acids that make up all the proteins in our body. And I think I mentioned earlier that the amino acids that we make are not necessarily the ones that bacteria make. So if we can figure out the difference between our amino acids and a bacteria's amino acid, we can make antibiotics to block their amino acids while not interfering with ours. So that's one of the thrusts of making antibiotics. Another thing, when we talk about cell membranes, when we talk about ribosomes, there are other ways that bacteria are different than us. And so we can target different things in those bacteria to make other antibiotics. So there are different antibiotics that attack bacteria in different ways, but you can do it through the amino acids. I do not expect you to memorize the 20 amino acids. I don't expect you to be able to draw the structure of them, but know that on one end is going to be the COOH, that carboxyl group we were talking about, and on the other end is going to be an amino group. So all of the amino acids have the nitrogen and the hydrogen and the carboxylic acid. And then they're going to have some other group. So cysteine is going to be important when we talk about how proteins fold, because sulfurs like to hang out with sulfurs. I always thought it was confusing because we talked about how the sugars go together and how you remove a molecule of water to do dehydration synthesis, and you do the same thing to join amino acids to make proteins. But for some reason, instead of calling it just dehydration synthesis, they call it peptide bond formation. So anyway, just thought I'd throw that in there. You take the OH off of the carboxyl group, you take the hydrogen off of the amino group, away goes your water, and now you have these two amino acids joined together. And there goes your water. So this is a covalent bond. Right there, sharing of electrons. But here, remember the word peptide bond instead of just covalent bond. It's still just a covalent bond, though. So the primary structure of a protein is which amino acids. So do I start with a glycine and then follow it with a serine? Which of the amino acids, in what order do I put them together? That determines which protein that I've made. But once I've made it and I've done the dehydration synthesis and done made the peptide bonds, now I need to fold the protein up so that it can perform whatever function it needs to do. So you have a secondary structure, and that would be like a Lay's potato chip. It's a beta pleated sheet. So it goes up, it goes down, it goes up, it goes down, just like a Lay's potato chip. Or wavy potato chip, if you use a different brand. You also have helix. So you can actually have it start corkscrewing and turn around and around, like the little ring that holds your 3-ring binder together. So you can have an alpha helix or a beta pleated sheet instead of just one long string of amino acids. And then it's going to fold back on itself. And if you have one of those amino acids that got a sulfur on it and it gets close to another one that has a sulfur, they'll start sharing the sulfurs. So they'll call it a disulfide bridge. And that's its tertiary, its third degree of folding. So one of the things that they used to say about on antihistamines is don't take the antihistamine if you are taking something like Synthroid. And I always thought that was odd. It's like, why can't I take an antihistamine? Or why can't I take a decongestant if I take Synthroid for lack of thyroid hormone? Well, come to find out, one of the things that a decongestant does is it breaks disulfide bridges. So all that snot and mucus is stuck up in your head and you can't get it out. If you take a decongestant, it'll break up the mucus so it runs out. So now you can breathe again. So you break up all that thick, yucky stuff and allow it to come out. But unfortunately, it'll also break disulfide bridges that you need, for example, in your thyroid hormone. So you always have to be careful with the medicines that you take because sometimes they have unexpected side effects because they affect something else besides just the snot and mucus in your nose and your respiratory tract. Denaturation is going against what is natural. So we know what the natural shape should be of various proteins, but if we apply too much heat, if we apply an acid or a base, then you're going to change the structure. So if it is permanent and irreversible, you can't get it back. So if you, in your mind, the example they always give you is crack an egg. And so you've got the yolk, and you've got that clear, sticky substance that's the white of the egg. If you were to put vinegar on it, if you were to put lemon juice on it, it would become white and milky, like in mayonnaise. But if you cook it with heat, it'll do the same thing. It becomes white, you can no longer see through it, and it actually kind of hardens because it cross-links. So it becomes opaque and stiff. So the protein that was in there is not natural anymore. It's not in its normal configuration. So it is denatured. A lot of proteins can change their shape to do their job, but they're not denatured. So enzymes are a classic example because they have to flex to pull molecules apart, or they flex to push molecules together, depending on if you're doing catabolism or anabolism. In muscle contraction, you're going to see these proteins moving and changing their configuration. When cells divide, you're going to see proteins changing their shape to help push the cell apart and make it into two cells. So proteins can reversibly change their shape, and as part of their job, that's what they do. But once you denature a protein, it can't ever go back to the way it was before. You're literally going to have to rip it apart, take the amino acids out, and rebuild it. So I talked about the natural shape of a protein, and I talked about disulfide bridges, where one sulfur is bonded to another sulfur and it holds it in a three-dimensional shape. Some proteins are globular. They literally look like a glob or a wad. And some of them are long, slender filaments, so we call them fibrous proteins. So you can make long, skinny proteins, or you can wad them up in a ball, depending on what you need them for. So examples of the fibrous protein would be your muscles. You have muscle fibers, and then your hair and your skin have fibers. So here's a slide that kind of puts together the primary structure is just how you put the amino acids, which amino acid follows which amino acid. And then the secondary structure is either the pleated sheet or the alpha helix, the coil. So that's your secondary structure. Your tertiary structure is where you have those disulfide bridges. So this folded over, and then that disulfide bridge is holding it in a loop right there. So you have the three-dimensional globular. This would be a globular instead of a fibrous. But then the quaternary, or the fourth level of protein, which we didn't talk about, is where you put several proteins together and they all work together as one unit. And the most classic example that I can give you is your hemoglobin. So you've got heme, you've got globin, and you're going to have 4. And inside you're going to have iron. You have these iron molecules that are able to carry your oxygen for you. So this is, hemoglobin is not a protein, it is 4 proteins hanging out together. The next few slides summarize some of the stuff that you use proteins for. The keratin that makes up our hair, our nails, and the outer waterproof layer of our skin. We have collagen, which is holding our skin and bones and teeth together. We use proteins as neurotransmitters, so they are able to send a message from one nerve to the next nerve, from the brain to the rest of the body, and so on. Some of the proteins act as hormones. So we have steroid hormones that are made out of cholesterol, like the estrogen, testosterone, progesterone, hydrocortisone. But then you have other hormones that are proteins, like insulin and thyroid hormone. So when we do the chapter on the endocrine system, we'll talk more about the fat-soluble or cholesterol-based and the water-soluble or the protein-based hormones. You've probably been hearing with COVID that there are receptors on the outside of our lung cells that are allowing the COVID virus inside of our lung cells and causing all the problems that we're having with COVID. Well, the protein that sticks up and invites the COVID in is made out of protein. So if we could block those receptors, then the COVID virus could not get inside of our cells. So that's one of the things they're looking at. The Inner Life of the Cell by Harvard, showing proteins, linear proteins holding cells together, receptors on the outside of cells, more of the linear, fibrous, globular, structural, the inner meshwork holding the cell together. Here you have self-assembling proteins, enzymes breaking down, microtubules being disassembled. And this is so cool, watching these motor proteins moving things. So definitely go watch that. It is cool. A lot of the proteins that we have require a cofactor or a coenzyme in order to work. So some of the cofactors are iron, copper, zinc, magnesium, calcium. So when they tell you that you need to eat things that have the proper vitamins and minerals, this is what they're talking about, because otherwise you can make the protein, but then it won't work because you don't have the cofactor. You don't have enough iron for your blood to carry oxygen. You don't have the copper that'll allow the enzyme to work. So you need a certain amount of this in your diet. And then we're going to talk about coenzymes, which are larger things, vitamins, nicotinamide adenine dinucleotide. How's that? We just call it NAD for short. And last but not least, the fourth group of macromolecules that you need to know about are your nucleic acids. So you have DNA and RNA, which are the nucleic acids that you're familiar with, but you should also know that ATP belongs to the group that it contains the nucleic acids. And when we talk about second messengers, we're going to talk about cyclic adenosine monophosphate, or CAMP for short. These nucleic acids have a sugar molecule. It's not glucose. It's either ribose or deoxyribose. And it's going to have a nitrogen-containing base. See, there's nitrogen, 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 nitrogen. And it's going to be one of 5 different nitrogen-containing bases. Adenine, guanine, cytosine, thymine, and uracil are the 4. So when we do the DNA replication chapter, we will talk more about these nitrogen-containing bases. But just for this, know that all the nucleic acids have a sugar, a nitrogen-containing part, and a phosphate part. Now this particular molecule is ATP, adenosine triphosphate. It has 3 phosphates. So we're going to find out how that's an energy molecule and how we need it in order for everything to function in our body. We have a whole chapter talking about how DNA makes up our genes and how our genes function and how we inherit various traits. And we're going to learn that there are 3 major types of RNA: the messenger RNA, the ribosomal RNA, and transfer RNA. So this is it for the chemistry chapter. I hope that I didn't go too fast and confuse you. I'll leave you with one last joke. This is what a student wrote on their paper when they were expected to tell what bonds with this and what was the product. And they just said, Hakuna Matata, which is no worries, take it easy.